Good afternoon. Welcome to everyone who is joining. It is exactly noon Eastern time, so we are going to get started. My name is Tamara Wyzanski, and I work in patient affairs at Sarepta Therapeutics. And the title of our presentation this afternoon is Introducing a New Limb-girdle Muscular Dystrophy Natural History Study. And our presentation today will run for about 30 minutes, and it will be myself and my colleague, Dr. Sarah Newhouse, who will be presenting during the 30 minutes, and then we will have a live Q&A at the end for the final 5 to 10 minutes, depending on the responses that we receive in the live chat. Next slide. So just a moment to thank the Limb-girdle community for tuning in, and also the Muscular Dystrophy Association, the MDA, for hosting us. We are really excited to go through the content today and thank them for the opportunity to host us on their webinar platform. So today, for our topics, we are going to be introducing our Limb-girdle Muscular Dystrophy programs. This will be Dr. Sarah Newhouse who runs through this and talks a little bit about our gene therapy platform. And then we'll get into the majority of the content for the half an hour, which is introducing our new natural history study. And we'll talk a little bit about the criteria to be able to enroll, the purpose of natural history studies, and just sort of run through what engagement and how to find out more information about the study will look like moving forward. I'll also wrap up with community resources at the end and talk a little bit about genetic testing and some more resources as well that are living on the limb-girdle.com site that is Sarepta sponsored for those that are looking to receive genetic testing and other resources for the community. And with that, I will turn it over to Dr. Sarah Newhouse. She's from our clinical development team, and she'll be leading the remainder of the webinar before the resources section. Thank you.
Thank you so much, Tamara. It's lovely to be here. It's exciting to meet with the community, and we are very excited to speak with you all today and share some information and learn from one another. So I am Sarah Newhouse. I am a pediatric neurologist. I have training in neuromuscular subspecialty medicine and have had an interest in research in gene therapy and understanding neuromuscular disease, rare neuromuscular genetic disease for over 5 years. I've joined Sarepta recently in February of 2020, and I'm very excited to be the medical lead and part of the clinical development team for this Limb-girdle platform moving forward. So to start today, we will speak to a general overview of the limb-girdle muscular dystrophies, which many of you are familiar with, and some of the key highlights on aspects we focus on at Sarepta. As we know, limb-girdle muscular dystrophy is a broad group of diseases with some very important common features. Although they can be inherited in a recessive or a dominant way, they can affect people of all genders and many ages. The disease can start at multiple times of life, sometimes early in childhood, sometimes early adulthood, and sometimes later on. We know that the commonality is that they're all because of a genetic misspelling or mutation in a gene, which essentially results in a misfunctioning protein, and sometimes even the absence of that protein altogether. Many of these proteins function to provide muscle strength, muscle integrity, and with those proteins misspelled or missing, it essentially leads to muscle weakness and progressive muscle weakness over time. So although there are a broad group of diseases that are what we say medically heterogeneous, which means mixed, the commonality is that it's a genetic root of disease, it's a protein misfunction, and the hope is if that's a mechanism we can repair, that would be a way to treat and improve these diseases. Many of them start with what we say proximal weakness, and that's the medical term for muscles that are close to our body, such as our shoulder or our hip girdles, meaning it's difficult to lift our arms over our head, lift heavy objects, or even get off of a chair or rise from our bed. So those are some of the common ways that these conditions present, and all of them also share a commonality of progressing over time. So from the time it starts, continuing to get worse over time at different levels. In some subtypes as well, the muscles that help us breathe, the respiratory muscles, and the muscles that help heart function, the cardiac muscle, can be affected. So each subtype is unique and specific, but those are some of the commonalities and areas we focus on. And I highlight those because those are a lot of what we hope to learn more about, to know where we can intervene, and to learn how the diseases progress in different subtypes. This leads us to our overall scientific approach known as gene therapy, also called gene transfer. The concept of this, I like to make an analogy of a sort of delivery truck. So depending on your limb-girdle subtype, we have a gene that has a mutation, and that is our gene of interest. That gene is created in a healthy way, also known as a transgene, and that's sort of the package, that's the valuable package. We're able to put that package in a delivery truck, which in this schematic is shown as our vector. So the vector is the delivery truck, the transgene is the package, and we hope to provide this, administer this to a patient, and allow it to be delivered to all cells of interest and deliver a healthy version of that transgene. As we know, there's much discussion of gene therapy for different types of diseases in the news and in the scientific literature. What's important to know about all of the limb-girdles is our target organ of interest, the area we're trying to improve, is skeletal muscle. And that's located throughout the body. That's not in one small area. So with this technology, our hope is to administer something that is wide-reaching and can reach the skeletal muscle throughout the body. And that's a brief overview of our gene transfer concept. A little bit more specifically, to get slightly more detailed, that gene transfer sort of delivery truck and the package is made up of three key components. As we mentioned, the vector functions as the vehicle, the way to deliver it to the body. And the transgene is the copy of the gene that can create healthy protein that can be delivered to the skeletal muscle cells. The promoter is the third part of that trifecta of these elements that work together, which functions to actually drive that transgene to make the proper protein in the cell of interest. So we sort of have the delivery, we have the package, and we have the element that increases that transgene production. And those are the three components of Sarepta's gene therapy engine. And so those are the key elements we think of when we're thinking of each limb-girdle subtype, each genetic disease, and how we're able to create a system where we can reach multiple diseases to benefit from this technological approach and this sort of platform. To that end, this is a slide showing the limb-girdle subtypes and this gene therapy sort of trifecta, this engine at work. If you can see by the color coding, the element that all of these gene therapy transfer mechanisms have in common is the delivery truck, the delivery vehicle. And in this case, we're using an adeno-associated virus, and the subtype here, it's AAV-RH74, which just is one of many subtypes of this kind of vector that is being used to deliver our transgene. Identified by the multiple colors, the red, yellow, green, and blue, are the various transgenes, sort of the packages that we're delivering. So depending on the limb-girdle muscular dystrophy subtype, and the root cause of that being a mutation in one of these genes, this is the healthy copy of that transgene to be delivered. And you'll see in the dark gray and light gray, the promoters that go along with that to help express that transgene in the tissues of interest. So I would highlight at the top, we have our limb-girdle 2D, E, and C, which are leading the way as sort of related group of muscular dystrophies, the sarcoglycanopathies, as they have a common mechanism of action and can be looked at as a group of diseases to treat in a similar way. And we hope to start with these subtypes of limb-girdle muscular dystrophy, learn as much as we can from this and the way they respond to this technology, and grow from there and expand. And so with that, I wanted to get into how we start the process of interacting with treatment and understanding the disease. And that starts with something very important called a natural history study. So for a moment, I'd like to introduce our natural history study in the limb-girdle muscular dystrophies, which has recently been titled Journey. This will be the name of our natural history, and I will get into more detail on that coming. Before we talk about ours in general, what is a natural history study? It sounds a bit of a curious word. The term natural history is a medical way of saying, how does any disease change and progress over time in different types of patients? So if we don't intervene with any medicine, how does a patient with a limb-girdle muscular dystrophy change over a period of 3 months, 6 months, 12 months, 10 years? What is the way that these diseases change over time in different types of patients? And until we are able to understand how the disease naturally progresses, we have a hard time understanding how our therapies can help and where they can intervene. And most importantly, as the patients with these various conditions, what are the most relevant symptoms to you as a person living with this, and where can we best help with our therapies? So the natural history study is really a foundation, a building block upon which we need to understand these diseases, to understand how we can build therapies and what is most important to patients and what areas we can intervene. So we understand on an individual and on a population basis how the disease progresses and under routine medical care, and really give a foundation for building on top with gene therapy. So I would say natural history studies are importantly looking at the disease in a more detailed way than you may do at a normal clinic visit. I'm sure many of you see your doctor every year, perhaps more than every year, and certain tests are done at those visits. The natural histories are designed to be a bit more detailed and systematic to look at every organ system over time. And I will use the next slide. What do I mean by that? When we talk about a condition like limb-girdle muscular dystrophy, there are multiple elements that can be affected. People's strength, people's energy, people's stamina, their breathing, their heart function, various elements can be affected. Typically, an annual clinic visit may not evaluate all of those systems in a standardized way. And what we do in a natural history is make sure at each visit we're looking at the strength, we're looking at your breathing and lung function, your heart function, and looking at your strength in a very calibrated way using physical therapy scales to get a sense of how that changes over time. Allowing us to measure these different components of the way that the limb-girdle muscular dystrophy presents, as clinical development and sort of drug developers, we work on something called an outcome measure. An outcome measure is a way to determine if our therapy has caused an improvement. So we start with the basis of understanding the disease. We start with understanding how your strength and function changes over time. We select a way to measure that, and then we can say, has our therapy intervened on strength, on your breathing, on your heart function? So working together with a natural history, we can develop an outcome measure in that area, sort of that organ system, and see how we can intervene. And so without knowing the way that disease progresses naturally over time, we have a really difficult challenge to find outcome measures that are meaningful to you and are valuable to the disease population, and to know how we can best intervene and measure the sign of progression and also measure the sign of improvement. So these are slightly more standardized, detailed tests that we would do in a natural history than you would have at a regular clinic visit. And we can talk a little bit about that as well. So in this initial natural history study, we are starting with the 3 sarcoglycanopathies that were mentioned in the beginning. We're starting with 3 subtypes of limb-girdle, LGMD 2E, 2D, and 2C. And the main goal of the natural history, as we mentioned, is to evaluate your motor strength, your mobility, your pulmonary and cardiac function, as well as some safety measures in labs over time, essentially in a 6-month to 3-year period, as long as you're able to participate. Data will be collected from each individual at set time points, and we will be able to contact you in between those time points to see how things are progressing. The last element of the overview is that we're very interested in understanding how the muscle functions and how the protein that is mutated has an impact on how you're doing and how you're feeling and your level of strength. So in the natural history study, slightly different than perhaps some others, we are actually requesting the ability to obtain a muscle biopsy as well as a skin biopsy. We would like to make the participation in this optional, and also if you've had a muscle biopsy in the last 10 years, if you are willing to share that muscle biopsy tissue with us, to be able to examine that biopsy and look under a microscope at your specific protein, at your specific muscle, and get an understanding of how the histology of the muscle biopsy compares to how you're doing, how you're functioning, and how you're feeling. And so we have to make that important link between how someone feels and functions and what their biopsy looks like under a microscope, which is a critical part of understanding how our gene therapy works. We're trying to get that delivery truck and that transgene to your skeletal muscle. And at the moment, the best way to do that is to look at that muscle under the microscope. And that's that key link between how you're feeling, how your disease may be changing over time, and how our gene therapy can be measured and understood. So some of the detailed tests that we mentioned, those outcome measures I mentioned before, things you may not do at every clinic visit. We have a number of sort of physical therapy, what we call motor outcomes or functional measures, meaning how well are you moving, how strong are you functioning. We do what we call a timed rise from the floor. So how quickly can you get from seated or lying on the ground to standing up. We time how quickly you can ascend 4 stairs, so 4-stair climb up and down. We do a 100-meter walk-run, which is a timed assessment of how quickly one can either walk or run, if able, 100 meters length of time. A dedicated physical therapy scale called the North Star Assessment for Dysferlinopathy, which you'll notice is one of the limb-girdle muscular dystrophies, but this scale is being used outside of just dysferlin for multiple limb-girdles, essentially looking at your neck strength, arm strength, shoulders, different tasks that we ask you to do in a very regimented sort of standardized physical therapy scale. You do that in a PT gym with the physical therapist. One of those tests is also called the PUL, or the PUL, the Performance of Upper Limb, which is a physical therapy scale just focusing on your upper extremities, your shoulder and hand and wrist strength, different sort of skills that you do to see how well your upper extremities are functioning. And then a pulmonary function test, looking at your breathing capacity and your vital capacity and elements of breathing. So those are some of the more standardized tests, not that everyone would do at a regular clinic visit, but that we would do in a regimented way in a natural history to understand how do those elements change over time, and what's the natural progression of those. So once again, to reiterate, our natural history, which has recently been titled Journey, and we want to review some of the eligibility criteria for you. As we mentioned, the subtypes we're focusing on for this initial part of the study are the subtypes 2E, D, and C. And we are accepting subjects of any gender, and the minimum age is 4. So anyone 4 and older. And you may say, why 4? Some of the physical therapy tests that we have are challenging for a child under 4 to follow those instructions. So they may be a certain level of detail or attention that has to be paid. And there are really certain skills developmentally, you need to be at least 4 years of age to be able to do those successfully. So patients under 4 are excluded, but there is no maximum age. So we are accepting patients of any age that have these subtypes that are willing to participate. And we understand people will have different levels of strength and function. And if there are tests that cannot be performed because of that, that's fine. That's why we've included a wide group of different assessments for people of various strength levels. We also require that you are under active care of by a neurologist or neuromuscular physician, someone that is able to provide routine health care for you so that we know you're being monitored, and should anything arise, you have a physician with you. Some of the exclusion criteria. So what does that mean? People that we wouldn't be able to include in the study. So if, for example, someone has any kind of cognitive delay or impairment that would compromise their motor ability. So this may refer to somebody with any kind of intellectual disability, anyone that would have difficulty consenting for themselves or following the instructions. As some of the scales are a bit complicated, we want to make sure they're understanding what we're asking and that their motor function isn't impacted by their ability essentially to understand the test, just basic understanding of the test. If you have an additional medical condition other than limb-girdle that may compromise your ability to comply, if there's something else seriously going on that has an impact on your ability to participate, that could be other conditions, surgeries, cancer, different things, but we would take that as a case-by-case basis. So the last part is if you are participating in an interventional study. So what does that mean? If you are a part of any kind of experimental therapy, or you're taking any experimental therapy, or you're part of a drug trial at another company, we would say that that's intervening in the natural course of the disease. And so you're sort of maybe taking or getting a medicine or intervention that isn't following the natural history. It could be changing that. So that would be an exclusion, as if you're on an interventional trial. But it's pretty open and pretty inclusive, and we're starting with those sarcoglycan groups, so we're hoping to reach as many patients as possible. We did want to mention that this trial is listed on clinicaltrials.gov. It's also listed, we also have members of our team and our group that are willing to answer questions and are willing to share information. We have a phone number listed here, as well as clinical trials at Sarepta.com, as well as our patient advocacy group at advocacy@Sarepta.com. If you yourself, a family member, someone you know would be interested, we would be very excited to answer questions. At the moment, we have one active site in the United States, and that is in Arkansas. And we are looking forward to expanding to other sites across the US and throughout, globally actually, as quickly as possible. And we hope to reach as many patients. We understand traveling can be a bit of a burden, so our goal is to open multiple sites to reduce that travel burden. I did also want to mention, we did a similar discussion, slightly longer talk for the Speak Foundation, one of the Limb-girdle foundations as well, about a little over a month ago on September 11th. At that time, we didn't have the study titled Journey, so we were referring to it as our natural history study, but it is the same study focusing on these three subtypes, where we do spend some time going into a bit more detail and answering a number of questions from the audience as well. So if you have any interest, please feel free to refer to that 60-minute Speak Foundation presentation as well. I think I mentioned the clinicaltrials.gov site as well. That is the identifying number there. But I think if you type in Sarepta and Limb-girdle, one of these elements will come up as well. So with that, I wanted to pass it back to my colleague Tamara, after just saying we can't express enough, although the natural history is not an interventional treatment trial, it really is an incredibly important contribution to the scientific community, to patients with limb-girdle, to patients currently and patients in the future. And without that, it is so challenging to move forward with drug development. It is a very incredible, valuable, and lasting way to contribute to scientific knowledge, to research, and to all of our efforts moving forward to create successful therapies and treatments in this subset of genetic disease. So we are so thankful to you if you're considering participating, and as the researchers and the teams that are thinking about these and working on them every day, we are very grateful to have you consider this study. So thank you very much, and I'll pass it back to Tamara for some additional information.
Great. Thank you so much, Sarah. I'm going to just chat a little bit about the limb-girdle.com resource and just social media for staying updated on general Sarepta updates. So limb-girdle.com, as I mentioned at the beginning of this webinar, is a Sarepta-sponsored site. It has information about free genetic testing resources. And I noticed a message come in the chat from Terry asking, you know, I've been diagnosed for 13 years. How do I find out my subtype? So that's a great question, Terry, and I would refer you and any others who have similar inquiries to refer back to limb-girdle.com. There's a tab at the top for the resources. We also do have an option to receive email updates. I will call out that this is for patients who are residing in the US. So we have different ways we can communicate with the patient community in the United States and outside the United States. But this is an opportunity for US patients to be on our listserv, where we'll be communicating updates through the rest of the year and into future years. If there are questions that we do not get to today, or there are things that are on your mind, or you want to connect to a live person from our team, either myself or anyone else I work with, I do encourage folks to reach out to advocacy@sarepta.com. This is an email that is managed by myself and our team, and we'd be happy to hear from you after today. And I wanted to call out social media. It's such a great avenue to reach people. We do have active channels on Facebook, LinkedIn, Twitter. And so this is a way, we have LinkedIn actually listed twice. So sorry about that. But we do you know, have these channels as a way to be able to communicate with us and see updates. So we invite you to follow us. We do have about 6 minutes, so I'm going to move right to the questions. And so we will take as many as we can. And before we do, I just wanted to take a note just on the last slide to really thank those who have already contributed to research. We know there are families who have done natural history studies in the past who have contributed to gene therapy research. So really just a note to say thank you for that time and energy. And we are so grateful to those families and the ones that will be in future studies to come. So one of the questions coming from the chat is, and this is for you, Sarah, is the study for those of us who are no longer able to stand or walk? So can you speak a little bit more to that about the criteria?
Absolutely, fantastic. So we are, as I mentioned, the age of 4 is the lower age. There's no upper limit of age. We do understand that a large percentage of the limb-girdle community is no longer walking full time, maybe in a transition mode using a mobility assist device. This is absolutely for you. Even if you can no longer stand or walk, we are able to assess your upper limb with some scales. We're able to assess your breathing. And most importantly, we're able to ask you questionnaires on how your life is impacted, and it would be incredibly valuable information. So the answer is yes, yes. There is no functional level, no low functional level, which we would not accept for the natural history study, just to be clear. Yes.
Great. Question, Sarah, about travel. So this question is, will we need to fly to Arkansas for this study? And if you can speak a little bit to how Sarepta may compensate those that need to travel either by plane, car, however they get to the sites, what that would look like for somebody who may enroll.
Absolutely. So Arkansas is the first site to be up and running, but we are expanding rapidly and aiming to have multiple sites available, not in every single state, but in major areas. So the hope would be, I don't know where you are geographically, but unless you're really close to Arkansas, you would not need to travel to Arkansas for the study. The hope is that we're going to be expanding very quickly to multiple metropolitan areas. If it's a situation that you would need to travel, the travel is absolutely compensated and reimbursed by Sarepta, so that would be covered. The goal would be to pick a site closest to you to minimize that travel burden, especially now in the times we're facing with the pandemic, and importantly, to minimize any stress or exhaustion on you. So we would say, stay tuned, reach out, let us know what state you're in if you're interested, let us see what site we're activating in your area, and try to get you connected with that medical group in the area.
Mm-hmm. And how often do you suggest those that are interested in seeing the updates check clinical trials or reach out to Sarepta? At what frequency should they anticipate updates?
Absolutely. So I think in general, depending on your specific limb-girdle subtype, checking clinicaltrials.gov to see what may be available, probably every 2 months is a good frequency to check clinicaltrials.gov to see if anything has been newly posted regarding the limb-girdle genetic diseases at large. I think for us, we're going to be making a large number of changes probably on a weekly basis. So I would say checking in with us once a month is a good amount of time to see which sites are up, which sites are new, and we're doing our best to keep the website updated as well. So I think for not just our study, but for the community at large, every few months, and then checking in with us once a month would be great to see when things are up and running. And we could maybe give you a specific answer depending on where you're located and what's going on there. Absolutely.
Great. Thank you. There is a question that asks, will there be a study for type 2J? And I know there are a lot of subtypes in the chat feed for those that Sarepta currently isn't in. So I'll just comment on that for a second, and then I'll turn it over to Sarah to add anything. You know, I think our programs right now are starting in these 6, and our natural history study is starting in the sarcoglycanopathies. But I'll just sort of echo what Sarah said earlier, that this research and the work we're doing, we hope informs future limb-girdle research across the board. So what we're learning now, and we're expanding our knowledge base now, we hope can grow research in other limb-girdles. I realize, you know, we don't have a natural history study right now for many of the subtypes of those that are on here. And you know, we're aware of that. And I think we're really hoping that the faster we can move this early research on, we are hopeful that there will be movement, not just among Sarepta, but other companies will start to look at limb-girdle and be interested in this. And the more activity and work that's done around this really lends itself to the work of researchers, the biotech and pharmaceutical company, and physicians and advocacy that are doing work around limb-girdle. Is there anything you want to add to that, Sarah, sort of around our other subtypes outside of the 6 that we're in, and sort of our plans or what this means for future development?
I think that was beautifully stated. I think it's a little bit of a chicken and an egg. The more interest there is, the more studies there are, the more interest it is. So the best way to get things going are to drum up that interest in the community and really sort of request that. I think we're starting, as we understand it's not covering all. We understand there are many. This is a single place to start, and we really hope to extrapolate any of these learnings as quickly as we can. Absolutely.
MDA has given us the liberty to go 5 minutes over, so we are going to do that because there are a lot of questions coming in.
Goodness, so great. Okay.
We'll end at 12:35. So let's do a couple more questions. When will there be plans to do a study for 2B subtypes or include them in natural history study? So are you able to comment on when Sarepta may move into natural history study for other subtypes that we're studying and moving gene therapy in?
Sure. So I think for the 6 that were listed, the top 3 are included as part of Journey at the moment, and the bottom 3 are the ones that are essentially on deck and next to be included and discussed. So once we have this successful model sort of up and running and we see the community engagement, those are sort of the next ones online. So we're very interested in learning. I'm sorry, did you ask for 2B specifically?
That was, yes, 2B was listed.
Okay, because we know there are also a number of other, with the Jane Foundation, other number of natural history studies ongoing, but I would say we are very eager to move into the subtypes to include all 6 on that slide. We're sort of starting with the top 3, but we're very interested. I don't know if I have a perfect time frame. I would like to say in the next, 6 to 12 months, looking at expanding to the remaining subtypes and getting people enrolled as quickly as we can.
Great, thank you. This question is about the actual time commitment to be in the natural history study. How often can the community, how often does one participate in the study? And I think I'm interpreting that right. So from a timing perspective, how long, how many years, or even how many hours can they expect visits to be? If you can speak about that a little bit.
Absolutely, fantastic. I think that was one of the slides that didn't make it. So in order for us to capture any progression of the disease, we made a decision based on literature research and talking to patients. We really want to see you every 3 months. So once you're enrolled at your ability, we'd like to see you in the clinic every 3 months, and we call you once a month in between to say, how are you doing? Anything changed? So it's a quick phone call in those in between months, and then it's a visit to the clinic every 3 months. During that visit, we would be doing the pulmonary function tests, and we would be doing the physical therapy tests, and you would maybe have some safety labs drawn depending on the visit. I would say typically it's at least for all of that to be done about half a day. So for you to have all the physical therapy measures done, meet with the physician, have a physical exam, if you need to have labs, and that's kind of allowing for time of transition. It's not that many hours of testing, but essentially sometimes about half a day or a day in clinic to get all of the tests done. That is correct. And the visit frequency would be every 3 months. And we're asking that you would, obviously when you participate in a trial, it's optional, so we cannot make you, but the study would be observing you for a period of 3 years as long as people participate. So it's a 3-year investigational study.
Great. And when Sarah was alluding earlier, sorry, go ahead.
Sorry, I'm so sorry. Every 3 months for the first year and 6 months thereafter. So it would be the most travel and visit burden the first year, and then importantly, it gets a little less frequent the last 2 years. Sorry, not 3 months for 3 years. It's a little less frequent in those last 2.
Yeah, no, that's a good point.
A lot of travel, we understand, yes.
Yeah, and I think the other piece, just around the slides that didn't make it into our 20-25 minutes today, the Speak Foundation did host us in September for a full 60-minute webinar. So that is on their website. It's also on their YouTube channel. So for those that want to see the visual of what Sarah was just talking about, I would encourage everybody to go back, scan through to get to the part where we talk about timing commitments. It's about halfway through, and that would be another way to kind of see and hear more about what you can expect for how long you can anticipate to be in the study, and also what is included in terms of phone call visits versus in person and things like that.
Perfect.
Another question, are there labs or blood work? And I think you alluded to that into what is required, but if you could speak a little bit more to that aspect.
Yes, absolutely. That's a great question. So as somebody actually mentioned previously, they said, I've had limb-girdle for 13 years. How do I know my subtype? And that is such a fantastic question. As you know, this is a cornucopia of different genetic diseases. And one of the key steps of being able to connect you in a perfect a treatment trial eventually is figuring out what genetic subtype you have. So as part of screening in the trial, we do safety labs, blood count, a chemistry panel, which looks at your electrolytes, those types of things. We also look at the muscle enzyme, CK, creatine kinase, which can be elevated in certain muscular dystrophies. And most importantly, prior to enrollment, we send specific genetic subtyping to make sure that the type that you've been told you have is consistent with the type you have, and we're able to look at that genetic information in detail. So I think there are some labs in the beginning for genetic confirmation, and then spaced throughout some safety labs to essentially look at the muscle enzyme over time, most primarily, to see how those things change.
Great. I want to do one last question before we wrap up, and I think it's really important, so it's a great one to end on. Can you speak to the muscle or needle biopsy that's needed, and a little bit around that piece of enrolling in the study?
Sure, absolutely. So as I mentioned, the need for the muscle biopsy is to better understand how a patient feels and functions and the strength that they have. As we know, some people are more quick to lose the ability to walk and more quickly to be needing a mobility device or a wheelchair, and others may walk for decades and have a different condition. So the best way for us to understand that is to look at the muscle under the microscope. And so at this moment in time, we can't get that information from a blood test or any other kind of saliva or urine or any other kind of liquid. The best way for us to do that is a muscle. There are different methods for which the biopsy can be obtained. Some of it is done with the surgeon doing what we call an open muscle biopsy, making a small incision and taking a piece out. There are other methods in which essentially a core needle device can be used, which is slightly more minimally invasive and can be almost used as taking sort of a round sample from the muscle and withdrawn. So I think depending on your site, there's different surgical expertise at different institutions. So without knowing specifically where, some surgeons do an open procedure, some surgeons can do a minimally invasive procedure. And I think our goal is to make this as minimally invasive as possible and as comfortable. We understand for children, it's painful, it's hard to hold still. Even for adults, it's also painful. So we want to make sure we can do this as minimally invasive as possible, but we do need to obtain a sample that is large enough that we can do certain types of testing on it for that protein quantification. So if you have had a biopsy in the last 5 or even 10 years, many times the institution that did your biopsy, maybe it was to determine your diagnosis. If you did the biopsy, that institution may have frozen tissue left, and with your consent, we may be able to use that frozen tissue from your previous diagnostic biopsy. So I would encourage, if the biopsy is a make or break, that would be something you would consider if you had a biopsy in the past. And obviously we're going to work to make it optional so that that doesn't deter you from participation. But there are a few ways to obtain the biopsy. And we can talk about that in more detail if we know what site you are. Yes.
Great. Thank you, Sarah. I want to end by thanking everyone for logging in this afternoon and taking the time to listen and send so many great questions. I realize we have to end, but please send us an email and then add yourself into updates for limb-girdle.com if you're living in the US. And we really hope to stay connected and also present at future opportunities about our work and stay connected with you all. So thank you again. Really appreciate everyone's engagement. Have a good afternoon.
Thank you so much.